Welcome to NanoString's webinar titled Unraveling the Complexities of Immuno-Oncology with 3D Biology Technology. Before we get started with our two speakers, I'll go through some housekeeping updates. The first one is if you cannot see the slide, please put in a request into the technical live chat. If you have questions through the webinar, feel free to use the Ask a Question feature to submit a question, and we'll make sure to answer that through the discussions that we have. If there's questions we have not gotten to, we'll make sure to give you a full answer after the webinar. With that said, let me now introduce you to our first speaker, who's Dr. Alessandra Fasano. She spent the last 2 decades focusing on clinical immuno-oncology research. She has an experience and passion for immuno-oncology research, and she's joined NanoString to really explore what optical barcodes can do in terms of answering some of the most interesting questions in clinical immuno-oncology. So with that said, I'm going to turn it over to Alessandra Fasano to start presenting on what's new and exciting in immuno-oncology.
Thank you, Niro, and good morning, everyone around the world, and thank you very much for joining this webinar today. My goal in the next 20 minutes are to briefly review our current understanding of the role of the immune system in cancer and how we translate this understanding in clinical effective treatments, at least for a subset of patients. Although at the same time, we've learned that we need novel biomarkers to guide both from a mechanistic point of view as well as a patient selection point of view and effective development and clinical implementation of novel immunotherapeutic approaches. So let's start since it's a big agenda. The immune system has the greater potential for a specific destruction of tumor cells with no toxicity of normal tissue and for long-term memory that can prevent cancer recurrence. And therefore, from this, it comes the promise of immunotherapy that is to really provide long-term cure. The last 30 years of immuno-oncology research have provided a very solid evidence that the tumors are recognized by the immune system and their development can be stopped or controlled through a process that we call immunosurveillance. So immunosurveillance is really a theory that the immune system is able to recognize and destroy tumor cells that are constantly arising during the life of the individual, and we call this elimination phase. However, in case when the immune system is not able to completely eliminate the tumor, for instance, the tumor becomes less immunogenic or the individual becomes less immunocompetent, a state of equilibrium development whereby the tumor does not progress, although is not completely eliminated. There is no clinical disease evident in this phase, like there is not in the elimination phase. This phase could be lifelong and thus mimicking the elimination phase, or can be disturbed by changes in the tumor or in the host, which allow the tumor to escape. So when the cancer cell resists or avoid or actively suppress an immune response, they really lead to tumor escape. To date, the most study of the immune system interaction has been performed in this particular phase, so the escape phase of immunosurveillance. So the tumor immune system interaction are shaped by different factors, and we can schematically, just for easy of thought, divide them or characterize them in 3 buckets: the host, the cancer, and the environmental factor. They all work together and converge to shape the interaction between the cancer and the immune system. So I will go through all these different buckets one by one. So let's start with the host. Well, the immune response in cancer has been described very elegantly by Aya Melman and David Chen as a cycle, the cancer immunity cycle. And this is really a series of carefully regulated events that can be self-propagating. Each step of this cycle requires the coordination of many factors, both stimulatory and inhibitory in nature. And this is the reason why an assessment of this process need to be done as a group, cannot be done singly. Let's go through the different steps. So the first step, neoantigens, they are created normally by the process of oncogenesis, are released by the tumor cells. And that's, sorry, there is a delay in the image. Pushing again. Okay. Sorry. Anyway, the first step, the neoantigens are released by the tumor cells and they are captured by the dendritic cells for processing. In order for this step really to provide an anti-cancer T-cell response, it must be come along with other signals done by pro-inflammatory cytokine or factors that are released by dying cells or by the gut microbiota, what we call Toll receptor agonists. In the next step that you see already now here, the dendritic cell is presenting the capture antigen on the MHC class 1 or class 2 molecule to the T cells, and this process results in the priming and activation of the effector cells against specific cancer antigens. Importantly, the nature of the immune response is determined at this stage. And really critical for the final outcome of the immune response is the ratio, the final ratio between T effectors and T regulatory. Finally, the activated T cell traffic through the vessel, and they go in the tumor bed, where they specifically recognize and bind the cancer cells through an interaction between the T cell receptor and is cognate bound to the MHC class 1, and then they kill the target cells. During the killing of the target cells, there is a release of other tumor antigen, and that's an important process that increases the breadth and the depth of the response, and start the subsequent revolution of the cycle. So in cancer patients, this cycle does not perform optimally. The tumor antigen might not be detected. The dendritic cell or the T cells can treat the antigen itself, and the T cell might also not properly home to the tumor, or factors in the microenvironment can suppress those effectors. So what is important is that the rate limiting step of any given patient can be different. So the goal of cancer immunotherapy is really to initiate or reiterate a self-sustained cycle of cancer immunity, enabling to amplify and propagate the cancer immune response without unleashing an unrestrained autoimmune response. But the most effective approach will be the one that involves selectively targeting the rate-limiting steps in any given patient. And this is where the need of biomarker is really stemming from. So let's look now at the factor at the level of the tumor that can shape the immune-tumor interaction. The genetic and cellular alterations that define cancer provide also the immune system with the antigens that generate and affect immune response. So essentially the antigens that allow the tumor to be recognized as known cells. And in this regard, association between a tumor mutational load and response to checkpoint inhibitor has been clinically demonstrated, really thus suggesting that a pre-existing adaptive immune response in a susceptible tumor is actually present. However, and very interesting recently, it has been demonstrated that the tumor oncogenic pathway can contribute to the immune evasion. And that is important because we will explain that 2/3 of the tumor in humans that do not show a pre-existing adaptive immune response. And so in this regard, the Gayesi group has identified a melanoma cell intrinsic oncogenic pathway, the Win-beta-catenin pathway, that contributes to the lack of T-cell infiltration in melanoma. Similarly, Patrick Yu's group showed that the tumor protein loss, which results in an activation of the PI3 kinase pathway, results also in a decrease in T-cell infiltration at the tumor sites. And finally, the group of Zhu et al. Provide evidence of a novel tumor immuno-evasion mechanism that are mediated by epigenetic events, so non-mutational events, such as epigenetic silencing of the Th1 type chemokine. Altogether, these new findings are important because they point to new candidates for immunopotentiation. So finally, the role of microbiota in the progression of extraintestinal tumor has only emerged recently, showing that the abundance of specific commensal bacterial species enhance the anti-cancer activity or immune checkpoint inhibitor by modulating systemic inflammation. However, a major question remains: how is these findings are relevant in humans? And I'm sure that a lot of new study will help us to understand that. So using gene expression signature or using actually immunohistochemistry, today we can identify two major tumor subsets in solid tumor. 1/3 of the tumor are the ones that they have a T-cell inflamed tumor microenvironment signature, meaning they have marker of T-cells infiltration, or they have a gene expression of a particularly functional signature associated with T-cell activation, and these are the ones that reveal a pre-existing adaptive immune response, thus suggesting that really the expression of local inhibitory factors are at play here. For this type of tumor are the ones that, if we look at the next slide, are probably likely to respond to checkpoint inhibitor that act at the level of the peripheral tolerance. The second group of tumors are the non-T-cell inflamed tumors that have no T-cell infiltration, and so they suggest that there is a lack of innate immune activation, or there is a problem in the T-cell trafficking, as discussed before, for instance, in the non-oncogenic pathway. So essentially, for this tumor, the approach from a therapeutic point of view will be to break central tolerance or to manipulate the oncogenic signal pathway, so repristinating T-cell trafficking. So the very different factors that can play in the cancer immunity cycle provide a wide range of potential targets, some of which have been successfully explored therapeutically, particularly the one antibody against anti-CTLA-4 and the anti-PD-1, PD-1 pathway. So what are these also called checkpoint inhibitors? The immune checkpoint receptor are crucial molecules that really are designed to fine-tune the immune response. Physiologically, the checkpoint signaling dampens the T-cell activation to avoid autoimmunity, excessive inflammation response. The checkpoint blockade therapeutically use monoclonal antibodies that release the brake from the suppressive cells, allowing them to activate and to recover their anti-tumor activity. Really, the renaissance of cancer immunotherapy began with the anti-CTLA-4 monoclonal antibody Ipilimumab in melanoma. CTLA-4 is a transmembrane protein that is expressed on the surface of T cells upon activation, and the binding of this molecule with the ligand results in a downstream signaling that culminates in T cell inhibition and decrease of cytokine production and proliferation. So importantly, the checkpoint function of CTLA-4 is the primary importance in the lymphoid compartments, and for most part is limited really to the activation of naive T cells. The anti-CTLA-4 antibody, Ipilimumab, was approved in March by the FDA in 2011 for advanced melanoma. And although the response rate was relatively modest, what was particularly striking was the fact that about 1/5 to 1/4 of the patients had very durable response. Really, the plateau of responses was at 3 years, stable at 10 years. This is the closest thing we have in oncology to a cure. In addition, more recently, the antibody has also been approved in the adjuvant setting. The second signaling axis that has been, used therapeutically is the one that consists of the PD-1 and this ligand, the PD-L1 and the PD-L2 this pathway is also a negative regulator, but of the immune response in the periphery. And so it's controlling the tissue damaging at the level of the periphery. So these explain in part why the immune adverse events of the blockage of this pathway are less severe of the one that the CTLA-4. However, what is important to highlight is that these two pathways represent two distinct non-redundant mechanisms of immune regulation. And so this is important because it's the base for the using combination of these. Currently, there are two FDA-approved anti-PD-1 monoclonal antibody, and while they are targeting the same pathway, there are many others in late-stage development that will become commercially available. So as I say, they are really targeting, they have the same mechanism of action, and they are targeting the same receptor, but they have currently different indication, and that is just reflecting the different clinical development plan. You can see the indication in the slide, and so I'm not going to go through that just for the sake of time. However, importantly is that despite the recent clinical success of the checkpoint inhibitor in a single agent and in combination in certain tumors outside melanoma in a certain subset of patients like Hodgkin disease, the majority of patients with metastatic solid tumor do not respond to checkpoint inhibitor, and although the combination are more effective, they are also more toxic and expensive. So the development and effective development of immunotherapeutic drugs require new biomarkers. However, because of the complexity of the immune response and of tumor biology, it is unlikely that any single biomarker will be very informative. To this regard, today, the majority of the work for the anti-PD-1 molecule has been done in evaluating at the level of the protein using immunohistochemistry, the PD-1. However, this is an issue. There are 7 drugs in development targeting this pathway, and currently, the current practice is independently for each of these companies really to develop their own assay, immunohistochemistry assay. And that is going to create a matrix of diagnostic and drugs that will result in challenging at the level of the clinical application. In addition, the PD-1 as also as a marker, as a single analyte marker, as a series of pitfalls, mainly in their cellular, spatial, and temporal heterogeneity, which contribute to the poor prediction accuracy of these biomarkers in the clinic. However, because of the complexity of the interaction that I just described between the tumor and the host and immune system, isn't likely they just looking at one analyte we will have very important information. And besides protein, the other 2 analytes that can be measured in the tumor and the host include the RNA in the form of gene expression profiling at the tumor or at the level of the immune system, as well as the DNA by measuring both the tumor molecular alteration as well maybe sequencing the diversity of the TCR region. And we have example of that. All exome sequencing on non-small cell lung cancer treated with pembrolizumab have shown association, as I mentioned before, between tumor burden and clinical benefit. But we have also examples of that in the gene expression profiling, in particularly examining the gene expression profiling in the tumor in relation of the clinical response, as shown by Dr. Ribas at ASCO. This year has shown that particularly signature, and particularly it was using a signature, a gene expression signature that was developed by the Merck scientists on the NanoString platform, showed that there was that signature was allow for an improved selection of patients that were likely to respond to anti-PD-1 therapy with pembrolizumab. One important was that this expression signature was not just tumor specific, but when was tested in a PD-1 positive head and neck squamous cell carcinoma show also a positive predictive value, and in particularly a much superior negative predictive value, thus suggesting a potential broad applicability of these new kind of biomarkers. So although the measurement of each of these different analytes is useful, the next generation of biomarkers will need to measure and integrate the complexity of the host, of the tumor, or the environment, and that's require measurement of different molecular entities, RNA, DNA, and protein, hopefully using the same sample so that to maximize the amount and the type of information that we get for sample, and also measuring the same units. So the ability of measure any combination of DNA, RNA, and protein from the same sample simultaneously on a single system is what we define NanoString 3D biology, and what we believe it is is the technology that provides the level of granularity that is needed for the next generation of highly predictive biomarker in immuno-oncology. And this level of granularity, as I say, is now achievable on the NanoString platform, and Dr. Joe Bisham will discuss in detail in the following presentation. So the ultimate goal, and this is one of my last slides, is really to make precision oncology not just a promise, as I started, but actually a reality through the development of an integrated immuno-oncology profile which measure and integrate information from relevant host, tumor biology, microbiota, and allowing in a decision tree manner to identify the ultimate limiting steps in the immune cycle so that we can properly match the drug mechanism of action with the tumor host biology at the level of the single patient. So I think I will stop here, and I will leave you just to read the last slide that is essentially reiterating what I actually say. I definitely just would like to highlight that we do believe the NanoString offered the tool to discover these novel biomarkers and to develop them into the next generation of immuno-oncology diagnostics, and I'm sure that Dr. Bisham will convince you of that.
Sandra, thank you for that fantastic presentation. In the space of immuno-oncology, discoveries are being made every day, so thanks for sharing some of your background in terms of how you see the space, how some of the therapeutic strategies are, as well as what the future holds for immuno-oncology. There are some questions online for you. We'll take a couple now before we go on to Joe's presentation. The first question is around, do you believe it will be possible to distinguish responder versus non-responding patients prospectively prior to immunotherapy through biomarker profiling? Will this be more difficult in patients who received prior treatment?
Well, I definitely think we are already doing some of this work. I think some of the data that I presented very quickly, but it is essentially suggesting that our understanding of the interaction between the tumor and the immune system, and also the effect that environmental factors like microbiota are having in this interaction, is increasing tremendously our understanding of what could be the different steps in which that are altered in the oncology. I definitely think this knowledge is ready to be translated practically in thank you to tools that, as I say, will allow to integrate this information so that we can do this in a more accurate way. We don't need to treat and expose patients to toxicity. We can combine treatment in a much more biology-based instead of the empirical base that we have today. I think it's going to be not the future of 20 years. It's going to happen very soon. Already we are making big steps toward that.
Alessandra, the next question is probably more dear to your heart regarding companion diagnostics. What do you think are some of the best practices for validating and developing prospective biomarkers into a companion diagnostic?
Yes, I think that's a very good question because, as I just mentioned, we know much more about the biology, but it's not only to understand what is the right biology that gives you, at the end, a diagnostic. It's really to the discipline and the knowledge and the capability of moving this science through a very regulated kind of environment and with high quality and standardization of process, so that these, and do that together with the pharma, so that you end up on the other side with the drug with the right companion diagnostic. So this cannot happen while the science happen in the academic side. So all these three steps are important: the science, the academia, the diagnostic company, as well as the pharma. We need all to sit at the same table and look at the problem from different aspects, but as a unified problem. Otherwise, we will end up with a lot of science that never really impact the patient.
Excellent. Thank you, Alessandra. For the remaining questions online, we'll make sure to get those answers to you after the webinar. At this time, we're going to move on to the second part of our talk, and I'd like to take a moment to introduce our next speaker, Dr. Joseph Beecham, who's the head of our R&D. Joe spent some time in academia, but then has spent well over the last decade developing high-end next-gen sequencing technologies. And he recently joined NanoString several years ago to really think about how to develop more capability out of barcoding technologies, optical barcoding technologies. He's the inventor of our 3D capability, and so today we'll talk a little bit about how 3D biology works and how we plan to use that to understand biology, especially in the space of immuno-oncology.
Hey, great to be here today, and Alessandra's great talk, and hope you get a feeling it's a real privilege for me to be able to work hand in hand with a CMO like Alessandra, because in that way, sort of my responsibility is the technology development, but it really has to be guided by the correct type of medical understanding of the problem. So, at NanoString, we work very closely together to really try to develop technologies that will actually help and translate some of these exciting discoveries that we make in science and actually do some good with them, actually get them to patients. So what I'm going to describe to you today is actually the development of technologies to try to work hand in hand with Alessandra so that we can develop these technologies that actually get applied in some realistic manner. What you see on this slide, and you're going to have to bear with me, the people on the phone, because I'm used to pointing at things, and who knows where the pointer is going to show up, but I'll give it a try. But I think the key thing here is that at NanoString, running R&D is great because I only have to solve one problem. It's how do we get more and more biological information out of smaller and smaller amounts of clinical samples. And our biological understanding is exploding, so we have to be able to follow that explosion in understanding with the right kind of biomarkers that can represent that information. So this is just a couple of slides on the introduction on how NanoString technology works. Key aspect about NanoString, this is technology developed by Lee Hood at the Institute of Systems Biology. And I'm sitting here in Seattle today. I can look out the window and see the ISB because that's why we're in Seattle. And these are the key developments related to development of single molecule counting optical barcodes. And the key thing I think for you to remember is that we multiplex up to 800 different analytes, and it's all digital detection. So you can't get more digital than counting a single molecule, and that's what these single molecule optical barcodes allow you to do. And I'll describe now the chemistry. So just to give you an idea on how the chemistry works and how you can interact with us as a company, you can sit at your desk, and what you see here are two reagents. One reagent is the optical barcode. The other reagent is what's called a capture probe. These are the things we make at our manufacturing site in Seattle, and that you can order up custom, or we have predefined panels for them. But just imagine you're sitting you're in your office, and you say, I want to measure the mRNA associated with, say, a key important immune regulator. Let's say you were interested in PD-1. So if you're interested in PD-1, I'm going to try to get this pointer thing to work. Who knows what will happen here? Let's see if a pointer shows up. What you see there is an optical barcode. And the optical barcode, you sit at your desk and say, okay, I'm going to dream up 50 base pairs. Actually, not dream it up. You would actually say, I'm interested in measuring PD-1. If I'm interested in measuring PD-1, what we actually do is you send us a gene name. And we will synthesize an optical barcode with a 50 base pair overhang, and that would be a hybridization target for, say, the mRNA for PD-1. And then say you're interested not just in PD-1, but you want to measure CTLA-4, OX40, PD-L2, PD-L1 all together. Then what we do is we synthesize one at a time hybridization targets for each one of those species. And what we do is we just move the little colors around on that optical barcode. So your CTLA-4 could start blue, yellow, and then when we make the 50 base pair hybridization target for, say, OX40, all we do is put the yellow ball first, the blue ball second, and then that represents the second target that you can measure. We make all those in our GMP facility here in Seattle, put them all together in a tube and generate multiplexed assays that you can run. So basically what you need to supply on your side of the equation is basically the raw nucleic acid that comes out of your sample. So you can see that there highlighted in red. You know, key aspect of our technology is we don't do amplification. We're not doing a whole bunch of cDNA conversions. We actually, all of our technology is hybridization driven. So we can measure the nucleic acids directly as they come out of the sample tissue of interest. And then the instrumentation, the automated instrumentation, just simply purifies this tripartite complex and does a single molecule counting. Now, when we work with protein technology, and this is a recently developed technology, what we did is actually set it up so that we label primary antibodies. You can see that on the left hand of the slide. We label primary antibodies with a small single-stranded 6-temer DNA. And then everything else, the workflow happens the same. So you actually interact with your sample just as if you're doing a flow cytometry experiment or a fluorescent immunohistochemistry experiment, except now when you get at the end, you don't go into a microscope or a flow cytometer. You can actually add your hybridization barcodes. They hybridize with the nucleic acid on the protein, and then you digitally count them, similar as the way you count nucleic acids. Now, you know, when I came to NanoString actually about 4 years ago, I'm sort of as guilty as everybody else. You know, I made genomic instrumentation or I made proteomics instrumentation and all these different things. You know, the fact is that that's not the way Mother Nature works. It actually pays no attention to these genomic boundaries or proteomic boundaries. It has a whole series of key molecular analytes that it makes. And the fact is that it utilizes all these different analyte classes together. So when I came over from Life Tech, you know, one of the key things that I was interested in doing was let's take these optical barcode technology and distribute it so that in a single simultaneous reaction, I want to be able to count DNA molecules, I want to be able to count the mRNA molecules, and I want to be able to count the protein molecules all simultaneously so I can actually understand the biology of the system in totality. And that's the key aspect and fundamental nature of our technology. This slide gives you an idea on how we accomplish this. That's the little sprint profiler. It's the benchtop encounter system on the right there. You can see up on the left side of the slide, you know, we start out with cells or tissue of interest. And what you can see is we start out and we need about 5 to 50 nanograms to do all of our DNA profiling. So this is if you're looking at copy number, single nucleotide variants, indels, things of that nature. Same cell pellet will be used. Actually, we'll use about 100 nanograms of the total RNA in there to actually monitor things like mRNA expression, microRNAs, so on and so forth. And then we'll use about a half a microgram to a 1 microgram of the protein to actually do the protein profiling. All of those together can be optically barcoded, counted out at the single molecule level in a completely digital manner. And that sort of forms the heart of what we call this 3D biology approach. What I'm showing you here is actually some of the very first raw data that we collected on 3D biology. See if I can get this cursor to work this time. Who knows whether it'll actually go where I'm pointing. But what we're actually looking at here is along the x-axis would be 5,000 cells worth of cell input. And then on the y-axis is 5,000 cells that represents a biological replicate. So this is the way we sort of debug all these assays. So a perfect biological replicate should have a slope equal 1.0. And what you can see when we do these kinds of experiments combining, in this particular case, we're doing a simultaneous analysis of 770 mRNAs. Those are all the little green data points. We're doing 15 proteins. Those are the orange boxes. And I got 5 gene fusion events in there, the purple triangles, 1 positive and 4 negatives. And again, I got them all analyzed, 5,000 cells in a tube, and you're getting a slope of biological reproducibility that's nearly, you can see, 0.98 out of a perfect 1.0. So with a single 5,000 cell input, I'm sort of getting the net accomplishment as if I've done a targeted RNA-seq experiment at about 250 to 500x. So if I ran mass spec on 15 different proteins and then actually set it up for about 5 different cytogenetics fish microscopes probing the gene fusion. But here we're just doing a single cell input on a single encounter system, all digital output. All the data can be plotted on a same graph. So we call this, when you actually put your digital data together like this and optically barcode all these individual components and allow you to combine that all together, we call that 3D biology. It's the ability to measure any combination of DNA and RNA protein on a single system. And I'll show you a number of important applications of this technology going forward. This technology basically solves a couple of major problems. One of them I denote, I call it the lost in translation problem. This is the problem I think I spent about 80% of my time when I was a professor at Vandy, actually trying to do translational research. I constantly hit this thing. It's like, well, my protein analysis doesn't match my RNA, and it's like, which measurement do I believe, and which one's right? I can't tell you how much time I wasted spending solving that problem. Similarly, I get my DNA doesn't match my protein analysis, and what do I do with that? When you collect data with 3D biology, you don't have to worry about that anymore. All the digital data comes out together so that if there is a disparity in what you measure, it's due to the biology. It's not because I don't have the right normalization factor for my mass spec data or something of some sort like that. Then it actually solves an additional problem. It's a different one, and this is really key. And if you're really developing translational assays, when you got to perform a bunch of different tests, what you find out is that, by the time I gave one FFP slice to the mass spec people, or maybe 5 of them to the mass spec and 10 of them to the cytogenetics people, I don't have enough tumor to go around to actually perform all these analysis. So then I have to decide who gets what. And what a terrible decision to have to make. With the 3D biology, you're never splitting up your sample. You get all that information at the same time, so you don't have to make those terrible decisions on how much sample goes to which measurement technique to answer what type of problem. Now, we developed this. I guess we launched this. I'm sitting here looking at Nero. He's sitting right next to me. We launched this RNA protein technology on September 11th of this last year. Actually, we're giving a talk actually at a high-rise in Manhattan on September 11th. It's a great time to be in a high-rise in Manhattan. But anyway, what you see here is that we developed this technology, and it's no good if it only works in my lab. I mean, it has to actually be totally reproducible. It has to work in your hands. So one of the key things that Nero set up was actually going out to these early access customers. Many of these customers are genomic-style customers. They hadn't measured a protein in like 20 years or something. We put this technology in their hands, and you can see how it works in their hands. So you can see the intra-user reproducibility, and that's just incredible. Look at how the mRNA protein replicates actually work when you're inside, say, one user doing it. And then we do something harder. Let's actually make 2 different users in the lab do the same thing. And what you see is you get exceptional reproducibility out of this technology. So it's not a technology that just works in my lab. It will work in your lab, and you'll be generating best-in-class reproducible data from these simultaneous multiplexed analyses. Okay, so I'm going to walk you through some case studies. Now, what I've been showing you is sort of like the analytical power of the technology. Now what I'm going to show you is that analytical power doesn't mean anything unless it can actually answer, interesting biological questions. So one of the first ones we looked at, we just don't go jumping immediately into a complex immuno-oncology problem to solve when we're developing this. So when I was a prof at Bandy, the lab right next to me is a guy that discovered some molecules called growth factors. And I did a bunch of work with this guy, Stan Cohen. He got the Nobel Prize for that. But we did a bunch of work together on understanding how a biological system responds to growth factors. So when we're debugging this 3D biology approach, what a great model system to use. Put a growth factor on a cell, and then watch the entire cascade of mRNA protein changes that happen after the epidermal growth factor binds to a cell. So what you see there on your right are data that comes directly out of some of the foundational panels we developed for studying cancer. In this case here, it's the cancer pathway panel there to your right. So when you actually do experiments with our, in this case here, it's a 770-plex that monitors all the key cancer driver pathways. We don't just give you raw numbers output. We give you those number output, and then we superimpose those digital mRNA results onto something that's interpretable at a pathway level, in this case here a KEGG map. So you can see every little red box means that when that cell was exposed to epidermal growth factor, everything in a red box is a digital mRNA that goes up, everything in a green box is a digital mRNA that goes down. And you'd be looking at basically 13 KEGG map pictures like this to monitor the response. So that's sort of what NanoString was always best in class at doing. But now we can actually, with 3D biology, we can actually examine the system at a whole nother level of detail. So what you see is all the great mRNA pathway-dependent results. That's what we've been giving groups, translational researchers and biomarker discoverers for a number of years now. But now what we're doing is superimposing on that mRNA results. You can see, let's draw that little square, the little circle there on your left that you see. That's the epidermal growth factor receptor. Now at the same time I'm getting all that mRNA result, you can follow that little arrow over to total protein and phosphoprotein signals. And you can actually look at the change in phosphorylation state of the epidermal growth factor receptor in real time as it responds to the EGF signaling. So you can see phospho-EGF go up, 15 minutes it spikes up, then you actually see it turns around and actually starts cycling back down. This is exactly the result me and Stan found at Bandy. It takes about 1 day for the phosphoreceptor to turn that signal off, come back to the surface, and then be ready to be charged again. So it's very nice to be able to see that the 3D biology could represent that result. I said, okay, that's still a little bit too easy. Let's go inside the cell and make sure we can actually probe the cytosolic protein changes at the same time. So we did that. So that's the second little ball you see there. Follow that logic through. In this case here, we're looking at phospho-ERK. Phospho-ERK, you can see, also spikes up at 15 minutes, shows this 1 day cycle time. But then also, see the little green data points I have here? This is the digital mRNA counts associated with one of the phosphatases that are going to take the phosphate off phospho-ERK, this thing called DUS-P10. You can see I can make that direct graph of mRNA and superimpose it directly on a protein. And I'm not doing some magic normalization here. This is the way the digital counts come out. I can plot those together in time. And I sort of call these 3D movies of these types of transitions. Okay, so I've talked a lot about how mRNA and protein works together. And then another development that's brand new that we're coming out with that I want to describe here is that we've enhanced and invented a new batch of optical barcodes that allow you to be able to probe single nucleotide variants at both the DNA level and the mRNA level. And we feel like this is very important because these single nucleotide variants are often linked to particular driver mutations. Or if you want to link back to Alessandra thought, this is sort of the private life of the tumor cells as they try to grow and escape the immune systems, oftentimes linked to these key single nucleotide variants. So we invented barcodes that actually have single nucleotide resolution, and in keeping with our philosophy, we measure it at the DNA level, which you see on the left, but we also measure it at the mRNA level that you see on the right. So every single nucleotide variant in DNA doesn't always show up in mRNA, so you want to be able to measure them in both places. And then in actuality, they don't always end up in the protein either. So 3D biology allows you to track them from one place to the next to the next. And we can do this tracking all the way down to very small amounts of tissue or cells that have the single nucleotide variant in it, but spec down to about 5%. And you can see we work in sort of best of class, 5 nanograms to 50 nanograms total input that we can resolve. these types of transitions. So again, what I'm doing here is sort of an intermediate step. We're going to sort of move from sort of a model growth factor system to something closer to immuno-oncology, and then I'm going to lead you into actually what some of this data looks like when it's embedded inside clinical trial work. So I'm going to go pretty quickly here. This gives you an idea. In melanoma, actually one of the key driver pathways is this BRAF V600E single nucleotide variant. So we're going to do a simple model system study. We're going to test for the presence of the single nucleotide variant. We're going to measure how the cancer mRNAs respond. We're going to measure how key IO proteins respond, all plus and minus a BRAF inhibitor. So you actually see a drug response superimposed on this entire profile. So I'm summarizing a bunch of data here. Just look towards the middle and the right columns. So the middle column of data, that is related to a case where we genotype, and that happens to be the wild type phenotype, if you will. And what you see there is you go right underneath the BRAF mutation graph, and then you look at that. What happened to all the mRNAs in those cancer pathways when you apply that BRAF inhibitor? Well, when you're a wild type, nothing happens. So you see that little volcano plot. Basically, no messages change greater than at a 50% confidence level in that entire cell system. We also looked at some of the key IO proteins on the cell surface. Same thing. You don't see anything that happens when you're a wild type genotype. You really don't respond to a BRAF inhibitor. But then you look over on the right side, you see, this case here, the genotype is a double mutant for V600E. That's what we measured. But then you look at that gene expression plus or minus that BRAF inhibitor. And you can see from that volcano plot, it literally is what it's called. It explodes with changes. And what you see is that you look at that response to that genotype, to that BRAF inhibitor, you can see all the explosion of the intracellular mRNAs. And you can map these by every pathway that's responded. That's all what's built into sort of the Encounter software associated with measuring these panels, so you can actually see the explosion and monitor every individual cancer driver pathways changing. Then on the bottom there, what you see is that what we're measuring here is actually an off-target effect. So these are changes on cell surface immuno-oncology proteins, HLA-DRA and CD73. And you can see that they actually are responded to a targeted inhibitor. I was a little worried about that when we first found it, but then when we actually went in the literature, flow cytometry had actually found one of these, I think CD73, and then qPCR had actually seen the HLA-DRA responding to vemurafenib treatment. So we basically discovered and was able to recapitulate, probably in a single experiment, one tech in about 3 days recapitulated, probably about 500 papers worth of experiments here. Okay, I'm going to sort of leave the land of sort of model systems behind. Before I do that, I just emphasize, there's nothing more frustrating when you're in the lab collecting data, to then be limited by having to toss that data over to some bioinformaticists that you may or may not be able to schedule and get time with them, and they may not understand your system and so on and so forth. So we actually spend a lot of time in R&D actually writing open source R statistical software that will allow you to be able to sit at your bench, you can have a technician, master, postdoc person that can actually accomplish about 90% of their data analysis sitting at their own desk. You can do that the day after you collect your data. And we felt that was very important. We spent a lot of time writing advanced software so that would empower you as the experimenter to actually be able to analyze and interpret your own data. So now I'm going to take you into some of the foundational panels that we set up to study cancer. And these are the types of foundational panels that are actually being put into exploratory immuno-oncology clinical trials. We basically set up a whole series of 770-plex assays that work on very small amounts of clinical sample material that measure on the left all the key cancer driver pathways. The one in the center is we worked with KOLs, and I'll describe to you how we did that. Special 770-plex for immune profiling, and then we also have another 770-plex linked to monitoring cancer progression and metastasis. These all work together. So as a foundational group, we can actually monitor the entire hallmarks of cancer series. We've been interacting with the experts in the field of immuno-oncology for a number of years, Probably about 3 years ago, we started interacting with a group called the Immunoscore Consortium, led by Jerome Gallon at INSERM in Paris. He did some of the really foundational work by flow sorting individual TILs, doing RNA-seq, figuring out all the RNA in them, generating RNA signatures. All that information became part of our foundational immuno-oncology panel that's available to you. What you see on the left on this slide is actually some of the original work that Jerome had accomplished. And each one of those little heat maps are RNA signatures unique to 24 different infiltrating immune cell types. All those RNA signatures are built into our immuno-oncology profiling panel that's available. And then on the right, you see what some of the data looks like. In this case here, it's Axel Husa's group at GSK. And what you're seeing there are NanoString digital responses. These are activated CD8s, activated CD4s. These are actually human-activated CD8s and 4s, and how they responded to BRAF and MEK inhibitors. And that's a paper you can reference and take a look at the raw NanoString data. This is actually one of our very first immuno-oncology products, and you can see launched in September 11th, 2015 in the high rise in Manhattan, if you will. And one of the key things we did is actually put some of the most important of the protein targets. This, as you can see on the left, the immunocycling that Alessandro very elegantly described, the entire immune cycle and how it progresses. We worked hand in hand with Alessandro. What you see on the right-hand side are digital optical barcoding assays that we developed for key protein targets as you walk around that immune cycle. And you can just go by the numbers here. It's a antigen presentation, that area 2. Well, we got CD4, CD40, and CD40L set up for that. And you can walk your way through there. The key thing is we got key digital protein-based assays that will work together with our mRNA assays to allow you to do some very advanced immune profiling. So now I'm going to walk you into some of our clinical trial work. You know, one of the first groups we interacted with on this 3D biology was essentially the father of protein biomarkers in cancer, a guy named Gordon Mills. Gordon Mills' lab did the entire RPPA project for TCGA. So all that data you see, the protein data is done with Gordon. We also work closely with Jim Allison, who he's one of the discoverers of CTLA-4, one of the biggest discoverers, just sort of broke open this entire IO area. And we're involved in interacting with them on their entire immunotherapy and targeted therapy clinical trial program. And we committed to IO, and I won't go through this in any kind of detail, but if you can follow for the last couple of years, one thing after another we're doing in this space. And you can see a nice quote from Gordon where he leads systems biology. He says, this 3D biology approach is the perfect way to actually monitor these types of systems. Okay, so how does it actually get used in discovery, IO discovery manner? This is what we got. So typically your samples come in 2 flavors. You typically have a day 0, perhaps an excisional biopsy, tumor excisional biopsy that will be profiling with their IO profile. You also have a longitudinal study with PBMC fractions, and we monitor the entire, both of these together. This gives you an idea of what some of our initial clinical trial work, this actually was our first one, where we're simultaneously monitoring. In this particular case, we're profiling the PBMC fractions. You can see the heat map in the lower left-hand side. Turned out the genetic content in our IO profiling has sufficient information so that it self-segregated in a non-supervised manner. It could distinguish the combination therapy arm from the monotherapy treatment arm. A very nice thing there, all I have to do is make a volcano plot you see on the lower right-hand side. In the lower right-hand side, discovered 7 biomarkers key to the combination therapy response. This is 3 days of experiments here, remember. Picked the samples up on a Tuesday morning. We had the 7 biomarkers on that Friday morning. We met with the PIs that were leading that study, showed them the 7 top biomarkers. We were blinded to the mechanism of action of the combination therapy. It turned out 6 of the 7 biomarkers there actually were linked directly to the mechanism of action that they unblinded for us after showing that result. So we have a number of examples like that. This is one we showed at SITC, a recent important Society for Immunotherapy in Cancer meeting in National Harbor, Maryland. We did just about a month ago or so. Again, another case where we're in the middle of a clinical trial, in this case, an IDO inhibitor. You can see on the left-hand side, we're doing full-blown IHC immunoscore type of monitoring. Then on the right, you can see the immune profiling from the NanoString technology. And there was a beautiful one-to-one correlation. High in IHC was high in our molecular profiling. Low in IHC was low in our molecular profiling. Of course, the molecular profiling gives you 770 answers, so all sorts of additional information you can glean from these immune profiling signatures. I'll show you one example. It's not done by us, so this tool is being put into, in this case here, it's a phase 2 clinical trial called CALM, and I'm just going to highlight what the raw data looks like so you can get a feeling. And just concentrate on the right-hand side. That's the raw biomarker discovery, targeted biomarker discovery data coming out of our immune profiling panel. You see the tumor, the original tumor on the far left, and you see high-resolution cell enumeration with multicolor IHC. Then those samples, those exact samples were put into our immune profiling, and the things you see up, so you see along the x-axis day 0, y-axis is day 8 treatment, and you can read the biomarkers directly off of the excursions above and below the slope equal 1 line there, and discover your biomarker signatures directly out of the immune profiling technology. SITC, there's about 14 posters presented. I just highlight this just to remember to tell you that. A lot of activities happening. This work just shows you that a company like Merck, for their key IO drug called Keytruda, developed NanoString signatures, and they announced at ASCO that they're interacting with us for clinical trial assay development work. This is a recent paper, literally came out just about a week ago, showing how our immune profiling along the y-axis actually maps in detail with digital pathology for both PD-L1 and CD8, a couple key markers, of course, and they're just using our immune profiling panel to get this information. There's no pathologist looking through a microscope to get this information in that particular case. This is my last slide. Essentially puts the whole thing together. We believe, like Alessandra described, that to be able to look at the DNA level, the mRNA level, and protein level are incredibly important. We understand this area of immuno-oncology. We're supporting it, and we're developing these tools to allow you to take and sort of be the first to invent a 3D signature that's going to be impactful in the area of immuno-oncology. These tools are out there. They're ready for use. And, I hope you can have some fun and actually do, great help. This immuno-oncology technology has such a powerful ability to help patients and affect patients' lives that we really need to get this technology out as quickly as possible. So these tools are designed to do just that. And I think we're in a good place to stop and take a series of Q&As here. Is that right, Nero?
Perfect. Thanks, Joe. Thanks for walking us through the 3D technology and explaining how it works and some of the key applications. We have a series of questions around the platform, the chemistry, the sample type, questions around concurrence. So we'll try and get through as many as we can in the time remaining. We will start off with a question on sample type. FFP continues to be the most challenging sample type. When you think about extracting DNA, RNA, protein information in a 3D biology context, how well do you think this assay will work with FFP samples?
Yeah, as you know, Nir, we're committed to clinical applications. You on clinical applications, you have to be able to measure on FFP. So what you'll see, the current commercially available RNA protein is actually 4 cells and dissociated cells, things you can run on a flow cytometer. In the first half of this year, you'll see this protein technology migrate right onto FFP. We are routinely doing multiplexed FFP on tissue slices of 10-plex. And if you come up to the upcoming AACR meeting, you'll actually see us show data 36-plex proteins on FFP. So totally committed to the space, and you'll see the protein and the mRNA technology and single nucleotide variant technology all mapped to FFP the first half of this year.
Joe, we have a question around the instrument capability of the nCounter, and the question is, has this instrument already been validated and approved for clinical use? And the tissue used is a biopsy after treatment. So the first question is around the use of the instrument in clinical applications, and the second is, again, sample compatibility.
Yeah, we have a whole line of instrumentation. We have a line of instrumentation, and we give them different names to them associated with this. So you have an instrument called the Flex instrument. The Flex instrument has actually been as a fully cleared FDA instrument. That's what we developed our in vitro diagnostic ProSigna assay, breast cancer assay, so on and so forth. So all this 3D chemistry works with all of our instrumentation. Our Flex instrument has been cleared for the FDA, so you can actually develop your assays, move them right to the clinic. And then we have this benchtop instrument that's called the Sprint Profiler. And that's a less expensive, low $150,000 box that can sit on your benchtop. That's not FDA cleared, but it allows you to have your own version. All the chemistry is identical, though, so whatever you find on the little benchtop instrument, you can immediately migrate over to the FDA cleared instrument.
Joe, I know we're up on time, but let's take one last question around the protein capability. There's a question about the process of probe hybridization compatible with the preservation of antibody-antigen interaction.
Yes, so what we do, and again, this depends, and we really follow the mechanisms associated with the protein side here. So if you have a set of antibodies, they've been cleared or validated for flow cytometry types of experimentation, we have 3D biology front ends that allow you to accomplish that in the antibody environment that it's used to working in a flow cytometry application and still give you your mRNA and your single nucleotide variant information from that same sample type. You happen to be working with an antibody that's cleared and validated for Western or for immunohistochemistry, then we allow that sample type to follow that particular protocol, and our mRNA and SNV work will follow that protein lead. So we develop these things so that they're all compatible, but we let the antibody actually be the one that we sort of, if you want to think about it, we serve the master, which is the protein antibody, and the mRNAs and SNVs we adapt that follow.
Great. Thank you, Joe. I know we're up on time. I want to thank everyone for joining this call. As I mentioned, there's more questions here than we've answered, so we'll make sure those questions are answered, and we'll get those answers out to you. We also want you to invite you to come and join our future webinars we'll have in the coming months. They'll focus on some of the new innovations that are going on in the space of immuno-oncology, but also some of the innovations that are happening in Joe's lab around 3D biology. And in the coming ones, we'll start to share some of the capability that we have around measuring DNA, RNA, and protein. With that, thank you very much, and have a good day.
So to Joe and Alessandra for their presentation. My name is Gary Geis, and I'm a principal scientist here at NanoString. I'm going to address a few additional questions that came in online, primarily around the 3D biology platform. So first, we have a question here about the CNV assay, the NanoString CNV assay. How large of a region can be assayed to measure copy number of unique regions? So if I understand this correctly, right, so we develop a custom platform for the detection of nucleic acids. So if you're interested in copy number variations, you can design probes to the genomic sequence. And of course, we have about, in our current system, we have about 800 probes that we can use to assess a particular region or regions within the genome. So it really depends on what sort of resolution you're looking like. For some of our panel products, we use 3 to 5 probes to cover a given gene. If you want higher resolution than that, of course, you can design probes across that cover the region more thoroughly. So it's really kind of up to the customer and how many genes and how many regions they can cover based on the resolution that you want. There is no limitation to the length of that region, just what the resolution is across that region. Okay, there's also a question here about how the 3D biology signature approach complements how the 3D biology signature approach complement and/or work with current flow and immunoassays in clinical trial applications. So I'm not sure exactly what you mean by that question, but essentially, what we believe is that you should be able to measure all three analytes on this system in a clinical environment. Of course, we're not directly the same as a flow cytometry or immunoassay, those approaches, but we are, we have the ability to measure proteins on an FFPE slide, for instance, and you can measure RNA from FFPE. So it should be able to fit into a clinical trial type of scenario, and that we would, obviously that would have to be clinically validated and go through the proper regulatory channels, and that has not happened for a 3D biology product as of today. I have a follow-up question about the CMV, asking about what about custom probes for the CMV, and of course, yes, we can do a custom probe. So for instance, you could design, if you so desired, if the region was large enough, all 800 probes to cover a big region of genomic DNA. So that would cover almost, let's say you spaced them every 1,000 base pairs or so, you could cover, almost a megabase in region. And if it were larger than that, you would obviously have to spread those out further. So again, our bioinformaticists can work with you to design, to answer the questions that you need to answer depending on the resolution that you desire for the CMV analysis. Okay, so with that, if there are no more questions, I'd like to, thank you for attending, and again, thank the presenters, and we look forward to hearing from you. Thank you so much.